Hello students, welcome back to Digital Learning DCE. Myself Dr. Manjula. In this session, let us discuss about bacterial plasmids and reproduction in case of bacteria by means of budding and fission. To start with bacterial plasmids, bacterial cells contain some non-essential extra chromosomal piece of DNA called plasmids. The plasmid is a short circular DNA molecule found inside the cytoplasm of bacteria whose size varies from 1 to 1000 kilobase pair. They carry genes for their own replication and also for antibiotic resistance. In this diagram, we can observe the two important parts like the bacterial DNA, which carries essential gene required for the bacteria and also for its replication, whereas the plasmids are extra chromosomal material which contain additional gene which are used under certain special conditions. The plasmids going to perform different types of functions. So based on the functions they are performed, they are categorized into different types which are as follows. Conjugative plasmids are the plasmids which contain tRNA gene and they perform the process of conjugation. They involve in the process of conjugation where the exchange of genetic material takes place between the donor and the recipient cell. F plasmid and R plasmid and also some col plasmids are examples of such plasmids. Non-conjugative plasmids are the plasmids which are incapable of initiating the process of conjugation. Example R plasmid and most col plasmids. Mobilized plasmids are those plasmids which are an intermediate class of plasmid which carry only a subset of genes required for transfer. Degradative plasmids are able to digest the unusual substances present inside the cell. Example, tall plasmid. Virulence plasmids are the plasmids which contain the genes of virulence which are able to cause disease in the host. F plasmids are those plasmids results in the expression of sex pili and involved in the transfer of genetic material during conjugation. Example of F plasmids are F plasmid of E. coli. R plasmids are the plasmids which contain genes for antibiotic resistance. Example PRP4. Col plasmids are those plasmids, they contain genes code for toxins to kill other bacteria. So example, col E1 is an example of col plasmid. Coming to the uses of plasmids. Plasmids play a very important role in genetic engineering since they are used as vectors or carrier molecules. They help the bacteria to survive stress conditions. Plasmids are also used in antibiotic resistance study. Some plasmids may contain genes that enhance the survival of an organism either by killing other organisms or by defending the host cell by producing toxins. Some plasmids also facilitate the process of replication in bacteria. So in this diagram, we can see the plasmids used as vectors in recombinant DNA technology where the plasmid from bacterial cell is isolated and then the DNA of desired sequence or DNA of our interest is going to be introduced into the plasmid so that it get converted into hybrid plasmid or recombinant plasmid which is going to reintroduced into the bacterial cell for further process during genetic engineering. So here we can find the R plasmid which is going to cause the resistance gene, hence it is used in resistance studies. Let us study reproduction in case of bacteria. As we all know, bacteria reproduce by means of asexual method, hence they have a dominant haploid phase. Bacteria reproduce asexually by two important methods like binary fission and budding. In case of binary fission, this is the most common method of multiplication observed in case of bacteria, where the parent cell divided into two daughter cells by forming a cell plate. So in this diagram we can see the steps involved in binary fission, where the bacterial cell, this is a mother cell, at the time of binary fission it enlarges in size, and the content of the cell, that is cytoplasm and also nuclear material, start distributing into two parts. Soon after, the nuclear material as well as cytoplasm divided into two, the cell invaginates, a constriction is going to be formed which extended to form a plate-like structure called cell plate. As soon as the cell plate is formed, this is going to divide into two bacterial cells. So, since the fission or splitting up of the cell takes place, the name is binary fission. As a result of splitting, two daughter cells are going to be formed, hence the name binary fission. We can observe this type of asexual reproduction in Bacillus subtilis and also in E. coli. Coming to the second type of asexual reproduction, that is budding. During budding, bacterial cell gives out a small outgrowth called as bud. This bud later filled with a bit of cytoplasm and a fragment of genetic material and also cell organelles. This bud later detaches from the parental cell by forming a constriction. This detached bud later grows into a new bacterial cell. We can see the steps involved in budding. At the time of budding, the parental cell start producing a small outgrowth called bud. In the second step, the content of the cell, both cytoplasm as well as the nucleus, start divided into two. Into this bud, a bit of nucleus along with the cytoplasm enters. Soon a constriction start to develop. When the constriction stretches, this bud separated from the parental cell. Here we can see the chains of bud formed, and the separated bud can lead into independent life. To sum up the session, plasmids is a short circular double-stranded DNA segment found inside the cytoplasm of bacteria, which are used as vectors in genetic engineering technique. Bacteria reproduce mainly by two methods that are asexual methods. One is binary fission and another one is budding. Binary fission is the common method of multiplication observed in case of bacteria, where parental cell divided into two daughter cells by forming a cell plate, whereas budding includes the formation of a small outgrowth called bud, into which a small amount of cytoplasm as well as nucleus moves and a constriction develops. Later, it detaches from the parental cell and grows into an individual new daughter bacteria. For further details about this topic, you can refer the following references. With this, I am going to conclude this session. Thank you.